Okay, so imagine two molecules. They are built from the exact same atoms in the exact same order. The only tiny difference? They're perfect mirror images of each other, like your left and right hand. So how on earth do you tell them apart? Today we're going to dive into a pretty cool scientific detective story about a new tool that could seriously change how we do things like develop medicines and even check our food for safety.
Right off the bat, you're probably thinking, so what? I mean, if they're made of the same stuff, what is the big deal? Well, it turns out that in biology and medicine, that tiny little difference isn't just a small detail. It can literally be the difference between a life-saving cure and a total tragedy.
And the stakes here are incredibly high. The thalidomide tragedy is a really powerful reminder of this. One version of that molecule, one mirror image, was a perfectly helpful sedative for pregnant women. Its twin, though, was a teratogen, which caused devastating birth defects. And that is exactly why being able to quickly and accurately tell which hand you're looking at is so critical.
Now, the sciencey term for these molecular twins is enantiomers. And just like your hands, they're mirror images, but you can't perfectly stack one right on top of the other. They just don't line up. These molecules are also called chiral, which actually comes from the Greek word for hand. So, how do we solve this puzzle of telling one hand from the other on a molecular level?
Well, that's the core challenge that researchers have been banging their heads against for decades. You see, the traditional ways of doing this can be really slow. They often need super expensive and complicated equipment, and a lot of the time, they just aren't sensitive enough. The world of science has been waiting for something simpler, faster, and way more efficient.
Now that's where this new breakthrough comes in. Researchers have developed a new tool, this tiny molecular probe they call SDHB. And it's so clever. It's designed from a molecule that's already easy to get, called S-BINOL, and its entire purpose is to solve this exact problem.
And what's really amazing is just how practical this thing is. First off, it's simple to make. We're talking one step. Then, you only need a teeny tiny amount, nanograms, on an electrode to get the sensor ready. And it's designed with these special arms, which have hydroxy groups on them, specifically built to, well, shake hands with other molecules.
So the huge question is, does it actually work? Well, to put it to the test, the scientists picked a target, tryptophan. It's an essential amino acid, super important for our bodies. And, just like the thalidomide, it comes in two mirror image forms. A left-handed version, L-tryptophan, and a right-handed one, D-tryptophan. First up, the control test. Using just a standard, off-the-shelf sensor, they measured the electrical signal from both versions of tryptophan. And as you can see from these totally overlapping peaks on the graph, the signals are identical. The standard sensor is completely blind. To it, they're the same exact molecule.
But now, look at this. This is the big aha moment. When they add their new SDCH probe to the mix and run the test again, everything changes. All of a sudden, that red peak for the D-tryptophan is way higher than the blue peak for L-tryptophan. The probe can absolutely see the difference. It's not even close.
And we're not talking about a small difference here. The peak electrical current for D-tryptophan was over 2.3 times higher than for its mirror image. That is a massive, unmistakable signal. The probe clearly has a favorite, and it's not shy about showing it.
Okay, so we know what it does, but the next question is how. How in the world can this tiny probe tell these two almost identical molecules apart so well? Well, it all comes down to a molecular handshake.
This is where we get to see what's happening. Using these really powerful computer simulations, the researchers could actually visualize the interaction. The probe forms these specific little connections called hydrogen bonds with each version. But the fit, that handshake, is different. The way it grips onto D-tryptophan on the left is just not the same as how it grips L-tryptophan on the right.
And the numbers totally back this up. The binding energy, which is just a measure of how strong that handshake is, it's stronger for D-tryptophan. See that more negative number, minus 31.76? That means a tighter, more stable connection. And that perfectly explains why the probe gives a bigger electrical signal for that version.
So to really prove this whole handshake idea, they ran a brilliant final test. They chemically blocked those key parts of the probe, those hydroxy groups, basically tying its hands behind its back. And the result? The probe with its hands free could tell the molecules apart, no problem. The blocked probe, it couldn't. This confirms it. Those hydrogen bonds are the secret sauce.
All right, so this all works great in a nice, clean lab. But what about the real world? Where things get messy. The researchers decided to put it to the ultimate test in a really complex substance: milk.
Yep, the researchers took a sample of regular cow's milk, which is a super complicated mix of proteins, fats, sugars, all sorts of stuff, and put their probe to the test. And what do you know? It worked perfectly. It could clearly pick out the tryptophan they added, which proves this thing is tough enough for real-world applications.
But you might wonder, could the probe get confused by other similar-looking molecules floating around? Well, as this chart shows, it's incredibly selective. It gives a huge signal for tryptophan while basically ignoring other common molecules like dopamine and histamine. It knows exactly what it's looking for.
And just how sensitive is it? Get this. The limit of detection was found to be 7.4 nanomolar. That is an absurdly tiny concentration, and it's significantly better than a lot of the other methods out there. So this probe isn't just smart, it's also extremely sensitive.
The researchers really summed it up perfectly themselves. This new method is a huge step forward because the probe is easy to make, it's easy to use, and it works with fantastic selectivity. It's just a really elegant solution to a very complex problem. And this really brings us to what's next. This breakthrough gives us a powerful new platform for spotting chirality. So if we can now easily tell the difference between the mirror images of an amino acid, what other critical molecules could we detect next? I mean, just imagine the possibilities for medicine, for food safety, for environmental science. The future of molecular detection just got a whole lot clearer.